Dear students, today we will see electrophoresis technique. So, with the help of electrophoresis technique, it is possible to separate amino acids. If it is DNA, then DNA sequence. So, with the help of southern blotting, we can study the fragments of DNA. If it is protein, when the protein is going to get separated, then we will get amino acid sequences, and with the help of amino acids, we can study each and every individual protein. So, in today's lecture, we will see in detail electrophoresis technique. So, electrophoresis is a method whereby charged molecules in solution, chiefly proteins and nucleic acids, migrate in response to an electrical field. Their rate of migration through the electrical field depends on the strength of the field, on the net charge, size and shape of the molecules, and also on the ionic strength, viscosity and temperature of the medium in which the molecules are moving. As an analytical tool, electrophoresis is a simple, rapid and highly sensitive. It can be used analytically to study the properties of a single charged species or mixture of molecules. It can also be used preparatively as a separating technique. Electrophoresis is usually done with gels formed in tubes, slabs or on a flat bed. In many electrophoresis units, the gel is mounted between two buffer chambers containing separate electrodes, anode and cathode, so that the only electrical connection between the two chambers is through the gel. In most electrophoresis units, the gel is mounted between two buffer chambers containing separate electrodes, so that the only electrical connection between the two chambers is through the gel. So, this is slab gel unit we are using for electrophoresis technique. This also one more slab gel unit, and this is the complete set of electrophoresis apparatus of slab gel unit, in which the portion for the valves, slab connection, power supply. So, this is called as flat bed unit. The net charge is determined by the pH of the medium. Proteins are amphoteric compound, that is, they contain both acidic as well as basic residues. Each protein has its own characteristic charge properties depending on the number and kind of amino acids carrying amino or carboxyl groups. Nucleic acids, unlike proteins, are not amphoteric, they remain negative at any pH used for electrophoresis. So, what is the role of solid support matrix? It inhibits convection and diffusion, which would otherwise impede separation of molecules. It allows a permanent record of results through staining after run. It can provide additional separation through molecular sieving. So, agarose and polyacrylamide. Although agarose and polyacrylamide differ greatly in their physical and chemical structures, they both make porous gels. A porous gel acts as a sieve by retarding or in some cases by completely obstructing the movement of macromolecules while allowing smaller molecules to migrate freely. By preparing a gel with a restrictive pore size, the operator can take advantage of molecular size differences among proteins. Because the pores of an agarose gel are large, agarose is used to separate macromolecules such as nucleic acids, large proteins and protein complexes, wherein polyacrylamide, which makes a small pore gel, is used to separate most proteins and small oligonucleotides. Both are relatively electrically neutral. Agarose is a highly purified uncharged polyacrylamide derived from agar. Agarose dissolves when added to boiling liquid. It remains in a liquid state until the temperature is lowered to about 40 degrees centigrade, at which point it gets. The pore size may be predetermined by adjusting the concentration of agarose in the gel. Agarose gel are fragile, however, they are actually hydrocolloids and they are held together by the formation of weak hydrogen and hydrophobic bonds. So this is Coomassie blue staining result. This also one more result where we have used silver staining. So what is meant by blotting? So blotting is used to transfer proteins or nucleic acid from a slab gel to a membrane such as nitrocellulose, nylon, DEAE or CM paper. The transfer of a sample can be done by capillary or Southern blotting for nucleic acids. So Southern in the year 1975 described the details about this blotting technique, or by electrophoresis proteins or nucleic acids. So with the help of this animation, we will see in detail how exactly electrophoresis apparatus works. Gel electrophoresis is a laboratory technique to separate large molecules such as DNA based upon their size by using electricity to carry them through a gel. Now a gel looks and feels like a piece of clear gelatin. It is made by dissolving agarose or another carbohydrate in boiling electrophoresis buffer solution. The solution is then poured into a casing tray to cool and solidify. The solidified gel matrix will have pores filled with the buffer solution. The DNA fragments travel through pores as the electric current moves through the gel. The larger the fragment, whether DNA, RNA or protein, the slower it will move through the gel's matrix. DNA can be separated by fragment size, with the smallest fragments traveling the furthest distances and the larger fragments travels to shortest distances through the gel. So this is a top view of gel electrophoresis, wherein these are the wells and this is the side view of wells. The wells of the gel will hold the sample of DNA. We can load the sample of DNA in the wells. Now for example, we will take 3 samples. First we will run sample A, then sample B, and simultaneously we can also able to run sample A and B, and then we will see what will happen. The gel is placed in a plexiglass box that holds the electrophoresis buffer and has wiring to conduct a current. DNA samples are loaded in the wells. So this is a side view wherein wells are there. In the center gel is there, then buffer solution, and this entire assembly is in plexiglass. So first we will run sample A. So the sample A has many copies of the same larger sized fragment of the DNA. Now we will take this larger size DNA with the help of pipette. So the pipetter draws up some of the sample. The sample is deposited into one of the wells of the gel. A source of electricity is connected to the gel box. This will provide a current that runs through the gel from the negative to the positive end. So this is connected with the electrodes. The electricity, when it is turned on, at the same time the DNA has a negative electrical charge. The current carries the fragments of DNA through the gel, weaving through the matrix of pores. Now we will take sample P. Sample P has many copies of two different size fragments of DNA, medium and small. With the help of micropipette, it draws some of the sample. We load this sample. The sample is deposited into one of the wells of the gel. A source of electricity is connected to the gel box. This will provide a current that runs through the gel from the negative to the positive end. The electricity is turned on. The DNA has negative electrical charge. The current carries the fragments of DNA through the gel, weaving through the matrix of the pores. The shorter fragment do less weaving, so they move faster through the gel. Now we will take sample A and B together. The sample A and B is a mixture of copies of the long fragments from sample A and the medium and short fragment from the sample B. The pipetter draws up some of the samples. The sample is deposited into one of the wells of the gel. A source of electricity is connected to the gel box. This will provide a current that runs through the gel from the negative to the positive end. The electricity is turned on. The shorter fragments do less weaving, so they move faster through the gel. Long fragments do the most weaving and move the slowest. So this is the complete mechanism where we can study the movement of fragments. All three samples are loaded into the wells and run through the gel. So all the fragments are separated. So with the help of this technique, we will be able to study either protein or DNA.